My name is James Watson, and I work at the Mahidol Oxford Research Unit, which is based in Bangkok, in Thailand. And this is a talk about recurrent Vivax malaria and trying to understand the cause of recurrent Vivax malaria. So just some background on MORU, or the Mahidol Oxford Research Unit. MORU has been based in Thailand for about 40 years now, and the headquarters is in Bangkok. I'm based on the Thai-Myanmar border at SMIU, and there are sort of study sites all across Southeast Asia and a study site in Kinshasa and DRC. And malaria is one of the big research areas that MORU focuses on, and one of the main things that MORU does is treatment trials, quite large treatment trials, and there's a big component of pharmacology, mathematical modeling, and epidemiology. So, Vivax malaria. About 100 years ago, Vivax malaria was the most geographically widespread cause of malarial illness, and it was pretty much everywhere in the inhabited world except in sub-Saharan Africa, where the Duffy negative gene or the Duffy negative allele has gone to fixation. And other than that, it was pretty much everywhere. And the most kind of unique characteristic of Vivax malaria is its ability to stay in the liver, to lie dormant in the liver, and then subsequently come out weeks to months later and give you a relapsing illness. So Vivax pretty much has exactly the same life cycle as falciparum malaria, which I'm sure most people are more familiar with falciparum than Vivax. And the thing that differentiates it from falciparum is this hypnozoite stage. So these dormant liver stages. And these hypnozoites can be dormant for a very long time, up to possibly 2 years. And relapse, so the waking up of these hypnozoites, has two major phenotypes. And the Vivax that was present in the sort of temperate parts of the world, so like the Netherlands, Korea, North America, you'd have this initial infection, illness, and then maybe you'd get your first relapse 9 months after that initial infection. Whereas in the tropics, so South America, India, South Asia, Oceania, these relapses occur much more frequently. There are more of them, and they're sometimes only, say, 3 or 4 weeks in between each episode. And the number of relapses that you might get from a single mosquito bite, the number of relapses you might get after having lived in an endemic area will depend on your exposure, which is a function of how many mosquito bites you get. So, for instance, here are some data on the left from soldiers, so US soldiers who have no immunity against Vivax malaria, who were fighting in the South Pacific, and when they came back, they got a lot of relapses, and some people were getting up to 15 relapses after coming back. And on the right here, I'm showing some data from about 72-year-old children who were followed in a refugee camp on the Thai-Myanmar border in the 1990s, and the green crosses shows each episode of Vivax malaria in each individual followed. The red triangles show mixed, and your falciparum are the circles, and you can see that some kids really have a lot of episodes one after the other, and that's more representative of kind of a typical scenario in sort of low transmission endemic area. So relapse is very important in Vivax, but after treatment with an effective blood stage drug, a recurrent infection could be caused by 3 different things. So if the drug doesn't manage to kill all of the parasites in the bloodstream, the remaining residual parasites can grow back to high densities and make you ill again. And this is called a recrudescence. So it's a failure of the blood stage drug. Or you could get a new mosquito bite, which causes a new infection, so it's called a reinfection. Or you could have a liver hypnozoite that activates, giving you a relapse. So there are three reasons for recurrent infection in an individual living in an endemic area. And the problem is, if you're following people over time, how do you distinguish relapse from reinfection and from recrudescence? And we propose to do this using the time to event, so the time between two consecutive infections, and the genetic signal of these paired infections. So the first question is, well, what do relapses look like genetically, and how do they look? Do they look any different from recrudescences and from reinfections? So suppose you're a person, you get bitten by a mosquito that's an infected mosquito. Here, this person's been drawn with a very large liver, and you can see that in the mosquito is injecting 5 sporozoites. So these are these circles here, and each color here represents a parasite lineage. And what's happened in the mosquito, so meiosis in malaria happens in the mosquito, so that sexual recombination happens in the mosquito. And here I'm showing 3 recombination events. One that's called an outcrossing event, so that's 2 genetically different parasite lineages that have recombined to give these recombinant progeny, so I have 4 progeny, 4 genetically distinct progeny. And then 2 what are called selfing events. So these are sexual recombination between 2 clones, which obviously gives you clones as the recombinant progeny. And this person is injected with some sporozoites, and then some of these sporozoites immediately, they don't go dormant, they immediately develop in the liver and cause an initial infection. And it's very important to understand that infections are generally a population of different parasites. And here you can see there's multiple colors in this initial infection. Now, recrudescence means that not all of these parasites were cleared by the drug. And here this is shown by this green lineage. There's one left over, it grows back, and it gives you a recrudescent infection, which is all green parasites. And so the genetic relationship you expect in a recrudescence is one that's clonal. So you expect to see exactly the same as one of the ones you saw before. So here it's green in the initial, and then green in the recrudescence. Now, relapse isn't coming from the previous blood stage infection, it's coming from the liver. So here, because you could have had the same ones being, the ones that woke up and the ones that are in the liver could be the same ones, you could see similar parasites, so either clonal parasites, or you could see ones that are coming from the same meiotic event. So here you could, this parasite here, which was sleeping and then woke up, has a sibling relationship with this other one that was one of the parasites that caused the initial infection. And so here the relationships you'd see are clonal as well, possibly, but also sibling relationships. But this relapse could also be caused by hypnozoites that were stored in the liver from before. And in that case, so this is a different parasite lineage, which is represented by a purple parasite. And so here the relapse could actually have new ones that you haven't seen before. And actually the whole relapse could just be caused by one single hypnozoite that wasn't present in the initial infection. So you could actually see no completely genetically different parasites in your recurrent infection from your initial infection. And this leads to the statement that the absence of evidence of relatedness between parasites in 2 infections doesn't mean it's not a relapse. It could still be a relapse. But also a reinfection, which is a new mosquito injects a genetically different parasite, causes a new infection, that also has the same relationship between the two infections. So that's to say they're strangers. They're not genetically similar. So in summary, a recrudescence, you expect that to be clonal. A relapse, you expect it to be either clonal or a sibling or a stranger. And a reinfection, you expect it to be a stranger. So the only relationship that's very specific to relapse is this sibling relationship. Now, what do we mean by relatedness? And relatedness, in the sense I'm using it, is this idea of probability of identity by descent. And you say that two alleles are identical by descent if they are descended from a common ancestor, which is not something you observe. It's something you can estimate with a probabilistic model. So, for example, imagine here you've got a short genetic sequence that is the same at two loci, so T and T and G and G. But as you can see from the two meiotic events that gave rise to these sequences, only the T actually comes from a common ancestor, and the G is the same, but it's just the same by chance. And so to estimate identity by descent, you need a probabilistic model that takes into account the background allele frequencies. That's basically taking into account the fact that you can have an identical genetic sequence by chance rather than by heredity. Okay, now what's the information in the time to event data? Now, relapse, both this long latency Vivax that was in the northern parts of the world and the frequent relapsing Vivax in the tropics, both of them exhibit very strong periodicity. So relapses don't seem to just pop up at random times. They actually seem to come at quite controlled and well-defined times. And reinfection is generally a seasonal event. And also the last thing to take into account is the drugs you give people to treat their malaria impacts when they get their next relapse or when they get their reinfection. So for instance, in the Sahel, you use long-lasting drugs like DHA proparaquine, which gives you a long-term prophylaxis. So if you treat someone with a drug that gives them long prophylaxis, you expect their relapse to come out much later. Now, the goal of this work is to estimate the probability that a recurrent Vivax infection is a relapse, and we use genetic data and time to event data, or time to recurrence data. And the data we have come from two large studies on the Thailand-Myanmar border, done between 2010 and 2014, and there's about 650 patients in each trial. In both trials, there was 1 year follow-up, and there was weekly and then monthly active follow-up in patients by looking at malaria, looking at slides, looking for malaria-positive slides. And so this is a lot of data where it's active follow-up, and every single infection, even if it's asymptomatic, was treated. Now, the model is quite complex, and it's sort of a model. It's a model of modules that are put together. So here, all the grey blobs are the models. So we have a model of allele frequency in this region. We have a model of time to event, and then we have a model that takes in both the estimates from the time to event model and the estimates of the allele frequency and the genetic data, and then gives you this final estimate of whether a recurrent infection is a relapse, a recrudescence, or a reinfection. So this is the sort of the flow diagram of the model and the data, and how they all put together. So the timing model is what's known as a mixture model. And so this says, well, sorry for the formatting hasn't come out very well, but this says every individual is going to recur at some point for some given reason, and that recurrence is either caused by a relapse, or it's caused by a reinfection, or it's caused by a recrudescence. and there are weights which determine how likely each of those events are. And so it's generating data from this kind of model. You can imagine flipping a coin, it falls on relapse, and if it falls on relapse, then you flip a coin that tells you when the relapse occurs. And here the weights for these different mixture elements in this model are parameterized as individual weights. So some people are more likely to have more relapses than other people. And relapse is broken down into a periodic element, so that happens at really a particular time, and a more late, what we call random, which is just an exponential distribution. And time to reinfection is an exponential distribution, time to recrudescence as well. And the novelty here is that it was specifically put in a periodic relapse element, which no one's really done before, and this, I think, makes it much more informative. Now, the genetic model is a bit more complicated. Now, you have, here on the right, I'm showing some example data from one individual in the clinical trial. And each time you see an infection, so there's the enrollment infection, and there are 2 recurrent infections. That infection will have a certain complexity. So that complexity means the number of different parasites that are present in that infection. And so first of all, you estimate the complexity of each of the infections. So how many different clones do I see? And then for those complexities, you put down a viable graph of relationships, both for the parasites within each infection and across each infection. So here I'm showing a possible relationship. So I'm saying this one and this one are clonal. This one and this one are siblings. This one are siblings. All of them have are strangers. So the absence of edge here means stranger. With all of the parasites here, and the 3 parasites present in the second recurrence are all siblings with respect to one another. So I'm not saying that is the relationship, but it's just one of the many possible graphs of relationships. And then you can, given that graph of relationships, you can calculate how likely you are to see those data that you've observed. And so here the data, I'm showing data from 3 microsatellites where the allele corresponds to a different color. So different colors, different alleles. And so you can kind of see here, well, these aren't exactly clones, because here you have a blue allele and here it's a kind of a pale yellow allele. But you can still calculate how likely you are to see those data given that graph. And you do this for all relationship graphs. And then different relationship graphs tell you different things about whether it's a relapse or a reinfection. So if you see a clone across 2 infections, that could be a recrudescence. If you see siblings, it has to be a relapse. Okay, so some results on our data. First of all, the time to event model. So this is fit to about 1,000 years patient years follow-up. And this is sort of the summary graph of what's the most likely cause of recurrence for a given number of months since the last episode. Now, green here corresponds to the individuals who were given primaquine. Now, I didn't really introduce this when I was introducing the clinical trial, but what we're interested in is comparing what happens when you give individuals primaquine, which is an anti-malarial drug, versus what happens when you don't give them primaquine. And primaquine is the drug that's the only drug available that actually kills these liver stages. So if you just give someone chloroquine or you just give them artesunate, that doesn't have any effect on the number of relapses. But if you give them primaquine, that targets the relapses. So if you give someone primaquine, then it doesn't have 100% efficacy, and they come back with a new infection a month or 2 later, then that has quite a high probability of being a relapse, maybe 1/3. but it still has a fairly high probability of being a reinfection. Now, if you don't give someone primaquine and they come back 1 month or 2 months later, so the pink line is artesunate monotherapy, the yellow line is chloroquine monotherapy, then that's almost certainly a relapse. And as time goes on, for the non-primaquine treated individuals, that probability of relapse goes down, and the probability of reinfection goes up. Okay. And what we could do is we have about 1,300 recurrent infections in the individuals who didn't get primaquine, and pretty much all of them, I mean, 90+% are the model saying these are very, very likely to be relapses because they're happening very soon after the previous infection, and they're happening with this quite clear periodicity. Whereas in the primaquine plus group, so these are the individuals who got primaquine, so who got this hypnozoite treatment, there's a sort of distinction between about maybe 10% that could be relapses, that have a reasonable probability of relapses, and the rest which have a very low probability of relapse. And note the y-axis scale here is on the log scale. Now, when you add in the genetic data, so as I showed you this kind of sequential model where you initially just look at the time to event data, and then those time to event estimates get fed into the genetic model, then it becomes quite clear the difference between those who didn't get primaquine and those who did get primaquine. So on average, in the ones who were typed, because not all the recurrent infections were genotyped, but about a third of them were. In the ones that were typed, if they didn't get primaquine, then almost certainly a relapse, and if they did get primaquine, almost certainly not a relapse. And what's interesting is there are two individuals here, well, two recurrences here. One of them had been treated previously with primaquine, one not, and about 10 months after their previous infection, they got an infection that must look genetically identical because it has a very high probability of being a relapse. Which says that, even in this area, this part of the world where it's thought that you don't really get long latency, you'd still get hypnozoites that are staying in the liver for a very long time before coming out. And this is just showing the data. So these are the individuals who both recurred and had recurrences that were genotyped, and the yellow here on the top are the ones who didn't get primaquine, the greens on the bottom, the ones who did get primaquine, and these black triangles here are failures, so it's either a relapse or it's a recrudescence. And the upside down open triangles are reinfections. And what you can see is pretty much everyone who gets a recurrent infection after taking primaquine, that's going to be a reinfection. And the ones who didn't take primaquine get a lot of relapses. And this kind of gives us this allows us to get a final estimate of the efficacy of high-dose primaquine, supervised high-dose primaquine, and the failure rate really in a 1-year follow-up is only about 3%, and this is quite a lot higher. Primaquine is basically a much more effective drug than previously thought, and the sort of in this epidemiological area, maybe 3 in 4 patients who don't get primaquine will have at least 1 relapse within a year, whereas only 1 in 40 will have 1 relapse if they are given primaquine. So it's a huge difference. It's a hugely efficacious drug if it's in this area at least. And it's important to note here that, as I showed at the beginning, the number of relapses you get depends on the epidemiology, and it depends on your exposure. So in areas of low transmission, people will generally have fewer relapses. And in areas of high transmission, people will have more relapses. So really the efficacy of a drug like Primaquine depends on the background epidemiology. So in this particular epidemiology, so it's at Taimyr in my border, you have a very low failure rate. But it's quite specific to this epidemiology. And so to summarize this, supervised Primaquine, very high efficacy, and you can use time to event data, genetic data, to sort of provide complementary information about whether a recurrent infection is a relapse or not. And this model made of modules kind of gives you this joint analysis in one framework that allows you to put together different data types. And the genetic model, as I've shown it, kind of applies to microsatellite data, but it's quite easily generalizable to different data types, like amplicon deep sequencing is one of them. And I'd like to thank you all for listening, and especially thank Amy Taylor, who I did this work with, who works at the Broad Institute in the Harvard School of Public Health, and Professor Nicholas White, who's my boss at Moru. Thank you very much.